You know the plastid, membrane bonded living bodies, smaller living bodies enclosed in the green eukaryotic cell. Eukaryotic cell. Such type of smallest living bodies enclosed in the eukaryotic cell known as plastids. So the plastid is smallest living bodies enclosed in the eukaryotic cell. Membrane bonded bodies enclosed in the eukaryotic cell. Commonly, plastid may be considered as prokaryotic cell associated with the eukaryotic cell. So plastid is smallest living body, smallest membrane bonded living bodies enclosed in the eukaryotic cell. Plastid only developed in the eukaryotic cell, not in the prokaryotic cell. Chloroplast, their number, their shape and size depends on the different group of green plant. In the algae, chloroplast may be spiral septa. In the Spirogyra, collar septa, star septa in the Zygnema green plant, green algae, and oval septa chloroplast commonly developed in the higher green plant. Their number depends on the location of the cell in the plant body. Greater number of chloroplasts developed in the cells which are located on the surface of the leaves or in the leaves, and less number of chloroplasts developed in the non-green part of the plant body. So the number depends on the exposure of the part of the plant body to the light. Chloroplast के number, ये light intensity और exposure of light पे depend रहता है. जो part ज़्यादा expose हो गया है light के लिए, तो वहाँ ज़्यादा number chloroplast के रहते हैं. उस part में, प्लांट के पार्ट में ज्यादा क्लोरोप्लास्ट के नंबर तैयार होते रहते हैं. सो अकॉर्डिंग टू द इंटेंसिटी ऑफ लाइट द क्लोरोप्लास्ट नंबर में बी इंक्रीज्ड. सो क्लोरोप्लास्ट इज सेमी एटोनॉमस सेल्फ डुप्लीकेटिंग सेल ऑर्गेनेल. Self-duplicating, semi-autonomous, green-colored organelle developed in the green plant cell. The chloroplast structure observed with the help of electron microscope. The structure of the cell organelle observed or developed with the help of electron microscope known as ultrastructure. So we have to study ultrastructure of chloroplast. Ultrastructure of chloroplast. Ultrastructure of chloroplast. Ultrastructure. Ultrastructure. Ultrastructure of the chloroplast shows different regions, different parts of the chloroplast. When the chloroplast observed under electron microscope, that may show structures. That structure is known as ultrastructure. Now here the ultrastructure of the chloroplast shows main part. First part is peristomium. Second part is grana. Third part is intergrana lamellae. Intergrana lamellae. And fourth part is stroma. So one by one we have to study in detail. Peristomium. First of all, we have to discuss about the peristomium. Peristomium. Peristomium is double membrane structure of the chloroplast. Double membrane structure of the chloroplast. This is the common shape of the chloroplast. This oval shaped chloroplast found in the green plant cell. Common shape of the chloroplast. The chloroplast is double membrane bounded cell organelle green plastid. It shows outer membrane and inner membrane. Outer membrane of the chloroplast and inner membrane of the chloroplast. This is the outer membrane. Outer membrane. Inner membrane. Inner membrane, outer membrane and inner membrane togetherly known as peristomium. The membrane of the chloroplast formed by lipid and protein. Now the space present between the membrane known as periplastidal space. Periplastidal space, or it is also known as intermembrane space, and it is filled by watery fluid. It is filled by watery fluid. It is non-colloidal in nature. So this membrane structure, double membrane structure known as peristomium. And what is the function of the peristomium? Peristomium is acting as semi-permeable membrane. We already studied the structure of the membrane of the cell. Fluid mosaic model we have already studied in the last period. So the peristomium acts as semi-permeable membrane. It allows the entry of the selected material inside or exit the selected material through the peristomium. Then another structure of the chloroplast is granum, or plural form grana. Grana. The number of grana are enclosed inside the peristomium. Stacks of lamellae, green lamellae, stacks of green lamellae suspended in the living liquid of the chloroplast are grana. Number of grana are group of stacks of lamellae, green stacks of lamellae suspended in the living liquid of the chloroplast is known as grana or are known as grana. Number of grana suspended in the liquid colloidal matrix of the chloroplast. Each grana is formed by stacks of thylakoid. Thylakoids. And these thylakoids, more than 2 thylakoids arranged one above the another and form a stacks of thylakoid known as granum. So number of grana are suspended in the living liquid of the chloroplast. The grana, each granum is formed by more than 2 thylakoids. Each thylakoid, when you observe under electron microscope structure of the thylakoid membrane, the membrane of the thylakoid is the location of the chlorophyll pigments, and chlorophyll pigment are located in the thylakoid membrane in the form of a structure or group of pigments, photosynthetic pigments located in the membrane of the thylakoid. Photosynthetic pigments located in the thylakoid membrane in a group, in a structure. That structure is known as quantasols. So quantasols are the light absorbing structures located in the membrane of the thylakoid of grana. So in the thylakoid membrane, as photosynthetic pigments are located that help in the absorption of light energy and conversion of light energy into ATP. So photophosphorylation process occur in the grana. That process is known as light reaction of photosynthesis occur in the quantasol, light reaction. So grana are the site of light reaction of photosynthesis. So function of the grana, absorption of light energy and conversion of light energy into ATP, that is known as light reaction. So grana are the sites of light reaction of photosynthesis. Another structure of the chloroplast is intergrana lamella or intergrana lamellae. The grana are interconnected by developing a membraned tubular known as intergrana lamellae, or it is also known as stroma lamellae. Stroma lamellae. These are colorless stroma lamellae or intergrana lamellae. Intergrana lamellae. The space present in the intergrana lamellae, that is known as Threads channel, and this colorless intergrana lamella developed or connect the grana of the chloroplast, and the intergrana lamellae are colorless double membrane tubular ultramicroscopic structure help in the fixation of carbon dioxide temporarily. The carbon dioxide primarily are first fixed by intergrana lamellae due to the presence of some of the enzymes. So, in the grana, light reaction of photosynthesis takes place, and in the intergrana lamellae, carbon dioxide temporarily fixed with the help of enzymes. So, enzymes present in the intergrana lamellae, and peristomium acts as transporting structure of the selected material inside of the chloroplast or outside of the chloroplast. Diffusion process, osmosis process occur in the peristomium, and in the grana, light reaction of photosynthesis takes place. Now, fourth part of the chloroplast is stroma. Stroma, liquid colloidal matrix present inside the chloroplast. The inner membrane of the chloroplast encloses liquid colloidal matrix of the chloroplast is known as stroma of the chloroplast. It is colloidal in nature because various types of components present in the liquid living material of the chloroplast. It contains RNA molecules, ribosomes, 70S ribosomes remain attached to the mRNA, 70S ribosome. RNA or mRNA remain attached by 70S ribosome, and the circular DNA may also present in the stroma of the chloroplast. Circular DNA. Circular DNA molecule may also present in the chloroplast, in the stroma of the chloroplast. The number of enzymes, protein molecules, biomolecules, number of biomolecules suspended or present in the stroma of the chloroplast. So it is colloidal in nature. And the number of enzymes are present in the stroma of the chloroplast that help in the dark reaction of photosynthesis. Dark reaction of photosynthesis. So, stroma is the site of dark reaction of photosynthesis. Grana are the sites of light reaction of photosynthesis, and intergrana lamellae help in the fixation of carbon dioxide temporarily. During that reaction of photosynthesis, the carbon dioxide fixed by intergranular lamellae released in the stroma that help in the dark reaction of photosynthesis. In the dark reaction of photosynthesis, glucose molecule carbohydrate synthesized. Now the structural component we have already studied of the chloroplast, that is peristromium, grana, intergranular lamellae and stroma. So here what happened, in the chloroplast, light energy absorbed by grana and converted into ATP and NADPH2. So light energy converted into chemical form, usable form of energy, and that usable form of energy, biological usable form of energy, is utilized in the dark reaction of photosynthesis for the synthesis of energy-rich carbohydrate, glucose molecule. So the chloroplast may also known as biological sugar industry. Biological sugar industry. Chloroplast may also regarded as biological sugar industry, because in this chloroplast, energy-rich carbohydrate glucose molecules are synthesized, and the glucose molecules may convert it into fructose. Glucose and fructose may interact and form sucrose. So here in the stroma of the chloroplast, during dark reaction of photosynthesis, the carbohydrate synthesized, and in the grana, light reaction of photosynthesis takes place, means photophosphorylation process occur in the grana. In the photophosphorylation, light energy converted into chemical energy, that is ATP and NADPH2. So assimilatory power synthesized in the grana of the chloroplast, and this power, usable form of energy, is used or utilized in the stroma during dark reaction of photosynthesis. सिंपल स्ट्रक्चर है ये क्लोरोप्लास्ट का, आउटर मेम्ब्रेन, इनर मेम्ब्रेन टुगेदरली नोन एज पेरिस्टोमियम. ये फर्स्ट स्ट्रक्चर है पेरिस्टोमियम. सेकंड स्ट्रक्चर that is grana of the chloroplast. Third structure intergranular lamellae, and fourth structure is stroma of the chloroplast. इस स्ट्रक्चर में, क्लोरोप्लास्ट स्ट्रक्चर में जो भी पार्ट दिखाई देते हैं, पेरिस्टोमियम, it acts as semi permeable membrane, outer membrane and inner membrane. So chloroplast is double membrane structure, and in the chloroplast the liquid living colloidal matrix is present, that is the stroma, liquid living material, where the process biochemical reaction occur, light energy not utilized in the stroma of the chloroplast. Hence it is known as dark reaction of photosynthesis. When there is no utilization of the light energy during photosynthesis and biochemical reaction taking place, known as dark reaction. And in the grana, light energy utilized during photosynthesis. The quantasols are the structures. Quantasols. Quanta means unit of light. Light energy absorbing structure. Quanta absorbing structures developed in the membrane, thylakoid membrane of the chloroplast. So the quantasols, number of quantasols developed in the thylakoid membrane and help in the absorption of light energy. In the quantasol, near about 250 to 300 photosynthetic pigments molecules are enclosed, help during photosynthesis. The chlorophyll pigment, chlorophyll A, chlorophyll B, xanthophyll, carotenoids, these xanthophyll, carotene, these photosynthetic pigments enclosed in the quantasome, and that help in the absorption of light energy and light reaction takes place. The special characteristics of the chloroplast is presence of circular DNA and presence of 70S ribosome. 70S ribosome. Presence of 70S ribosome. 70S ribosome. 70S ribosome and circular DNA. Circular DNA. Circular DNA is the most important characteristics of prokaryotic cell. So chloroplast regarded as prokaryotic cell associated with the eukaryotic cell. So chloroplast shows symbiotic association with the eukaryotic cell because the chloroplast shows the characteristics of prokaryotic cell, that is presence of circular DNA and presence of 70S ribosome. The 70S ribosome formed by 50S and 30S subunits. 50S and 30S subunits form 70S ribosome that help in the synthesis of protein with the help of mRNA and various components required for protein synthesis, and circular DNA. So this circular DNA may act as a mini chromosome. It's chromosome of the chloroplast. Chromosome of the chloroplast, circular DNA. So independently, protein synthesis and metabolic activities occur during division of the chloroplast. So chloroplast is semi-autonomous, self-duplicating, semi-autonomous, self-duplicating cell organelle, double membrane cell organelle. Such type of ultrastructure of the chloroplast observed under electron microscope. ये क्लोरोप्लास्ट का स्ट्रक्चर 3 मार्क के लिए या 2 मार्क के लिए फिगर में asked, and the explanation with figure may ask for 5 marks. So in detail we have studied the structure of the chloroplast. ये वीडियो आपको अच्छा लगा तो लाइक एंड सब्सक्राइब करना. थैंक यू.